Well, hello everybody, and welcome to the middle lecture of Unit 2. We're going to continue talking about experimental techniques in molecular biology and build on some of the concepts that we talked about in our first lecture of this unit, namely PCR. So here we'll be talking about gene cloning specifically, and every other subtopic will relate to cloning genes. So that's going to include vectors or plasmids. These are the circles of DNA that we clone genes into. Restriction enzymes, the molecular scalpels that we use to cut the DNA sequences we wish to clone and get them ready to paste or ligate them into these plasmids. And then we'll move on to studying gene expression in two experimental techniques. We'll talk about reporter genes to study single gene expression, and microarrays to study gene expression on a more global or genomic scale. So let's get right to it. Constructing completely novel DNA molecules is not something we could do about half a century ago, and now it's commonplace. In even the simplest, smallest labs across the world, we can clone or isolate and characterize essentially any DNA molecule or gene that we wish to. These are called recombinant DNA molecules. The textbook definition of recombinant DNA is DNA molecules or sequences that are not normally or naturally occurring, and they're made artificially in a lab. More specifically, recombinant DNA molecules involve isolating specific DNA molecules, mostly genes, most of the time genes, and moving them into another DNA context or another sequence that we can either manipulate or study more easily. So often we make these recombinant DNA molecules by taking two different pieces of DNA that weren't naturally found together and forcing them to exist together in one continuous DNA sequence. We glue them together with an enzyme called ligase. And these DNA sequences that we're gluing or ligating together can be sequences from different organisms. They can artificially be synthesized, so a unique DNA sequence that's never even found in nature. The sky's the limit here. And by using techniques such as PCR, which we've already learned about, and restriction enzyme digests, cloning, things that we'll be talking about today, really the sky is the limit in terms of what DNA molecules we can build and the types of experimental questions we can not only ask, but we can also answer. So at its crudest consideration, this is really what we mean by making recombinant DNA molecules. Again, we have this circle of DNA that we call a plasmid or a vector. Almost all of the recombinant DNA we make will be in this circularized form and will exist in the context of a plasmid. We can take different DNA strands, molecules, or sequences and clone them or glue them together into whatever sequence we want. So here we're making a kind of a Frankenstein, a recombinant DNA molecule that is made up of three different inserts or three different DNA sequences from three different sources. And the end result is this single continuous DNA sequence, this single gene perhaps, that's really made up of three different sources. Again, we can create this DNA essentially from scratch. So doing this allows us to custom build DNA that can contain whatever DNA properties we wish to study or manipulate. So perhaps it contains desired mutations. We want to study the effects of those mutations in a model organism, such as yeast or mouse. Maybe we add visible tags to the ultimate protein that this DNA gene, this recombinant gene, will express. Maybe we make this protein more detectable in some other assay. So again, we can think very, very big and kind of create whatever DNA molecule and presumably whatever protein, if that DNA molecule is an expressible gene, that we wish for our experimental purposes. So let's build the individual parts that we need to do this kind of work, and then we'll bring it together near the middle of the lecture into the idea of gene cloning. So first, of course, we want to have some background on plasmids or vectors. Again, these are the circles of DNA that we will most often put our cloned genes or DNA into. So plasmids are circles. They contain a very large region that we can place our DNA into. That's going to rely on these restriction enzymes that we'll be talking about in just a moment. And these plasmids also contain an origin of replication, labeled ORI here, O-R-I. These origins of replication allow the plasmid to be copied or replicated in the host organism that will manufacture it for us. And they typically have some kind of trackable marker, some way that we can discern and differentiate between host cells that contain our plasmid and host cells that do not. Here we see the trackable marker on this plasmid is an ampicillin resistance gene. So this gene confers ampicillin resistance to the bacterial organism that carries our plasmid. So if we expose bacteria to our plasmid. Some of the bacteria will absorb our plasmid and carry it. Those are the bacteria that we want. Other bacteria will not successfully absorb the plasmid. That's a failed process. We don't want those bacteria. So we can expose bacteria to our plasmid and then grow that bacteria on ampicillin. All of the bacteria that receive this plasmid have the ampicillin-resistant gene. When they express that gene, they will have the ampicillin-resistant phenotype, and they will be able to grow in the presence of ampicillin. All of the bacteria that don't get our plasmid will continue to be ampicillin-sensitive, and they'll die on media that contains ampicillin. So these trackable markers that are present in plasmids allow us, again, to separate what we want, cells that contain the plasmid, from what we don't want, cells that do not contain the plasmid. So again, we've got the origin of replication, which allows replication to occur. Essentially, the origin of replication is the place at which the cell can separate the two strands of DNA, using each strand as a template to engage in replication. We don't really talk about the process of replication in this course. That's more of a cell biology topic, but we've covered similar topics. Transcription is essentially the copying of a DNA gene into an RNA form. So just like we had to create a transcription bubble at the promoter, we have to create a replication bubble here at the origin of replication. And also PCR is essentially in vitro DNA replication. It's DNA replication in a tube, so the principles of PCR pertain quite a bit to replication. So that's what the origin of replication does. And again, the ampicillin resistant is the trackable marker. We also have specific plasmids called shuttle vectors. For those of you that are engaged in undergraduate research or might be engaged in undergraduate research in the future, you might use a shuttle vector. Just like shuttles move between two locations, like the airport shuttle moves between the parking lot and the airport terminal, shuttle vectors can move between two host organisms. The most common shuttle vector is a bacteria yeast shuttle vector. So this is a plasmid that can exist in bacterial cells, usually E. coli, and it can also exist and be maintained by yeast cells. So here we see that we have an origin of replication for bacteria, as well as an origin of replication for yeast. Eukaryotic and prokaryotic origins of replication are very different, and so we need one for each host cell. We've got a trackable marker for E. coli, again, an antibiotic resistance gene, and we have a trackable marker so that we can differentiate yeast cells that have our plasmid from those that don't. That we see this leucine biosynthesis gene. And then we have a place to put the DNA that we wish to clone. That's this multiple cloning site that we'll come back to in just a second. And in order to make sure that the yeast believes that this plasmid is a chromosome, we can actually give it a eukaryotic centromere. We put the centromeric sequences in the plasmid itself, and that fools the yeast cell into thinking that this plasmid is a chromosome, and it will distribute it efficiently during mitosis and manipulate it as though it was a chromosome. So as a general overview of cloning, now that we have some sense of what a plasmid is, we have our circular DNA of our plasmid. We have our origin of replication, our selectable marker, and our region that we can put the DNA into. And then we have the DNA fragment which we wish to clone. This is our PCR amplified or our isolated DNA. And so somehow we're going to cut the plasmid. That's where these restriction enzymes come in. We'll also cut the ends of our DNA to be cloned, and then we can enzymatically glue those molecules together, inserting our DNA of interest into the plasmid. And this is our recombinant plasmid now. This is two different pieces of DNA that don't naturally go together, but they go together because we made them go together. And again, this is our recombinant molecule. We can then do a transformation, which is the term used for getting foreign DNA to be absorbed by a host cell. So we would take bacterial cells such as E. coli, we would mix them with our recombinant plasmid. Some of those bacterial cells would be successfully transformed. They would absorb our plasmid. Others would not be transformed. They don't get our plasmid, and that's where the selection comes in. Again, we would take those cells and we would plate them all on media containing ampicillin in this case, and only the cells that contain our plasmid would be able to grow in the presence of that antibiotic. Cells that do not get our plasmid don't have the ampicillin resistance property, and they would die on those ampicillin-resistant plates. So a very straightforward, a very common workflow for DNA cloning and replication of that recombinant plasmid. And we can get more complex. We won't really talk about the additional complexities around this here. The scope of this course doesn't really go that high, but we can do this with a variety of different plasmids. So we can actually transform multiple plasmids into E. coli cells, and we can actually create what are called DNA libraries. We can have entire genomes represented on thousands and thousands of independent plasmids, and we can transform those plasmids into host cells. So I just wanted to introduce this so that you have this notion that it's not always just one plasmid per cell. We can get multiple plasmids transformed into cells if we plan the experiment properly. So what are these restriction enzymes? Let's talk a little bit more about what these DNA cutting molecular scalpels do. So restriction enzymes do two main things. They bind to specific DNA sequences. So they bind with specificity, meaning they recognize a unique sequence of DNA. The most commonly used restriction enzymes bind to 6 mers, 6 base pairs of DNA. And then number 2, they cut that DNA. Where they cut that DNA differs depending on the restriction enzyme. We see that some are what we refer to as blunt end cutters. They cut that 6 base pair sequence right in the middle. And others are sticky end or offset cutters. They cut in an offset way. And we'll talk about some of the implications of these different cut patterns in just a moment. So again, restriction enzymes recognize a specific, usually 6 base pair sequence of DNA. They bind to that DNA with specificity, and they cut that DNA sequence as well. I want you to also take a close look at these restriction enzyme binding sites. Again, these 4 sites that we see on the screen are 4 recognition or binding sequences for 4 different restriction enzymes. And take a really close look at them. See if you see anything that is unique. And I'll even give you time if you'd like. You can pause the video and just take a close look at these. Look for patterns. Look for things that are a little bit interesting here. And what you may have noticed is that these DNA sequences of these restriction enzymes read the same 5' to 3' on each strand. So here, the recognition sequence is GTT-AAC, 5' to 3'. And if you look at the opposing strand, and if we carefully read it 5' to 3', we see that it's GTT-AAC, same exact sequence. And we see that's true for all of these, AAG-CTT, AAG, CTT, CTG, CAG, CTG, CAG, and finally GAA, TTC, GAA, TTC. This is unique in DNA in that we have this internal complementarity. In other words, we have this mirroring effect where these 6 bases read on the top strand, 5' to 3', is the identical sequence to these bases read on the bottom strand, 5' to 3'. And we call this property in molecular biology being a palindrome. So in the English language, a palindrome is a word or a phrase that reads the same forward and backwards. One of the common examples is race car. If you take a moment and you write the word race car down on a piece of paper, RACECAR, that reads race car right to left, just like it reads race car left to right. That's a palindrome in the English language. These are molecular palindromes. They read the same 5' to 3' on the top strand as they read 5' to 3' on the bottom strand. Now, there are major implications there, and they explain the cutting pattern that we see. For example, the recognition site for HindE3, the enzyme that is represented in the upper right, the recognition site, the DNA sequence that HindE3 binds to and cuts is AAG-CTT, and it specifically cuts between the A's. But you'll notice that HindE3 then has the ability to bind to the top strand, and it recognizes AAG-CTT, and it cuts between the A's. But it also recognizes and binds to the bottom strand, AAG-CTT, and it cuts between the A's. Similarly, HEPA1, binds to GTT-AAC and cuts between the T and the A, but it's going to do the same exact thing on the bottom strand and cut between the T and the A. In this way, because of these palindromic sites, restriction enzymes of this type create what we call double-strand breaks. They don't just nick or cut one strand, they shear the DNA molecule into 2 fragments because they're cutting on both strands in a very similar location. And that's because of this palindromic nature of the recognition sites. Because these restriction enzymes bind to both the top and bottom strand and cut both the top and bottom strand, we get this shearing or fragmenting of these DNA molecules. So here's EchoR1 again. It's a very, very common, perhaps the most commonly used restriction enzyme. And again, it binds to and cuts GAATTC, cuts between the G and the A. We see that EcoR1 is binding to the top strand and cutting between the G and the A. It's binding to the bottom strand and cutting between the G and the A. And if we separate or denature, disanneal those two strands, we see that we are left over with sticky ends. Any restriction enzymes that cut in this staggered way are going to leave these sticky ends. Sticky ends are portions or single-stranded overhangs of cut restriction enzyme sites that are hungry for or ready to base pair to their complementary sequence. I should also point out that this entire process of restriction enzyme cutting is not an artificial process. This isn't something that bioengineers created from scratch. This is a naturally existing system. Fascinatingly, perhaps, I think it's fascinating, the major source of infection and natural death or infectious death in bacterial cells is viral infection. There's an entire class of viruses called bacteriophage that specifically infect and kill bacterial cells. So that's bacteria's number one kind of enemy or risk, is being infected with viruses. And not surprisingly, perhaps, bacteria have evolved defense mechanisms, or you might even say immune systems, to protect against viral infection. And that's exactly what these restriction enzymes are. Restriction enzymes are the bacterial defense against viral infection. So when a virus infects a bacterial cell, it injects its DNA genome into the bacterial cell, and that viral genome hijacks and takes over the bacterial cell and ultimately kills it through a process of infection called the lytic cycle. If the bacteria has the ability to recognize and destroy that viral genome before that viral genome has an opportunity to hijack the bacterial cell, the bacteria will essentially be protected from the viral infection. It can cure itself of the virus. And that's exactly what restriction enzymes evolved to do. They bind to and cut foreign DNA so that that foreign DNA cannot launch an infection. That viral DNA cannot launch an infection. Now, that's all fine and good, but then, of course, the bacteria runs the risk of cleaving its own DNA, fragmenting its own genome. Just like our immune system had to evolve a way to recognize self as self and not attack ourselves, autoimmune diseases show us the danger of having the immune system turn upon ourselves. So too did this restriction enzyme system have to evolve a mechanism to recognize bacterial DNA as self-DNA. And that's through what is referred to as a modification system. So these bacteria also contain DNA methylases that add methyl groups specifically to restriction enzyme sites, and any methylated restriction enzyme site is not cut by the enzyme. So now we can see how this system works. E. coli, the bacterial cell, methylates all of its DNA, and all of that DNA is then recognized as self and is not cut by the restriction enzyme. Here comes a bacteriophage, a virus that infects the E. coli cell. It injects its viral genome as the first step of that infection, and that viral DNA is not methylated. The restriction enzyme recognizes that viral DNA and cuts it up into pieces, neutralizing the infection, and the bacteria is cured of the virus. So really, really nice system. I bring this up because this idea of methylated DNA can be a pain in the rear for molecular biologists, because if you happen to be working with methylated DNA and you want to cut that DNA with restriction enzymes, you can't because it's protected. So you have to demethylate that DNA before it can be cut. So just a little side point here. But let's return to this idea of sticky ends. So when we cut with these off-center staggered cutting restriction enzymes such as EcoR1, when we pull the DNA apart after the double-strand break, we have these single-stranded sticky ends, and that is what's going to allow us to clone the DNA sequences that we wish to copy. We have a huge array of restriction enzymes that we can use. This is just a very, very small subset of the restriction enzymes that are available to be purchased. Some of them are blunt end cutters. They cut right in the middle of that sequence, so we don't get sticky ends. Others are offset cutters, so we get sticky ends. We see EcoR1 here, again, the most common. Hindi3 we've talked about already. BAMH1 is another very common one. TAC. So this is TAC's restriction enzyme, not its polymerase. We can get a restriction enzyme from TAC. So what you might be picking up is that these funky names, NOT1, Hindi3, EcoR1, they are actually representative of the host organism from which we stole this restriction enzyme. So the restriction enzyme EcoR1 is called ECO because it comes from E. coli. Hindi-3 comes from Haemophilus influenza. TAC, of course, comes from Thermus aquaticus. So we pay homage to the host organism that we stole these restriction enzymes from by naming the enzymes after those organisms. So we pick restriction enzymes that have recognition sequences that match the application we're trying to achieve. We've got convenient sites in our DNA of interest. We're going to choose restriction enzymes that will cut at those sites. And then we want to clone or insert that DNA into our plasmid, and that's where the sticky ends are going to come in. If we cut our DNA of interest with the same restriction enzyme that we cut the plasmid, we will generate complementary sticky ends. Here we see a sticky end, TTAA, and one of these 4 sticky ends is complementary to it. See if you can spot which one. Which one of these 4 sticky ends in black is going to base pair with the sticky end that we've created in red. And it's this third one here. We see that this adenine is going to base pair with this thymine, this adenine with this thymine with this adenine, this thymine with this adenine. So if we can imagine that this sequence floats in and base pairs with this sequence, we get nice, perfect complementarity across those sticky ends. And that's how we get these puzzle pieces of DNA regions to fit together. And we have to glue them into place with ligase, but it's that fitting that's important. So these sticky ends allow for a very specific, the analogy I always use is almost like a jigsaw assembly, this very specific insertion and recognition of our DNA of interest with our plasmid. And it's achieved simply by cutting the DNA of interest and the plasmid with the same restriction enzymes. So now we can put a little bit more meat on the bones of cloning. Again, we have our plasmid here. Maybe in green, we have the antibiotic resistance gene, the tracking gene. Maybe here is our origin of replication. And this is our multiple cloning site. This is that region that we're going to insert our DNA of interest into. And now I can tell you that what makes this a multiple cloning site, what makes this a region that we can insert DNA into, is that it just has dozens of different restriction enzyme sites in it. It's got an EcoR1 site, a BAMH1 site, it's got the TAC site, it's got SAL1, it's got dozens of restriction enzymes. So it's almost like a Swiss Army knife, an all-purpose insertion site that allows you to use whatever restriction enzymes you need to for your DNA of interest. So maybe we cut the plasmid with EcoR1 and BAMH1, so we leave an EcoR1 sticky end on one end and a BAMH1 sticky end on the other. We cut our target gene of interest with the same restriction enzymes, EcoR1 and BAMH1. We leave those sticky ends. And now the EcoR1 sticky ends will have perfect complementarity on this side. The BAMH1 sticky ends will have perfect complementarity on this side. And this DNA will fit perfectly into the gap of the plasmid. If we add a little bit of ligase, we'll seal that DNA in place. We'll make new phosphodiester bonds across those splice junctions or those cut junctions. And we'll have created a whole continuous new recombinant plasmid that contains our gene of interest. So let's see that with the sequences in place, again, using EcoR1 and BAMH1. So here we can see the sequences that are left, those sticky ends. Again, for EcoR1, our sticky end is AATT on our gene of interest, and the BAMH1 sticky end is CTAG on our gene of interest. So here is us cutting our gene of interest, GOI, our gene of interest, away from its native context, wherever it is normally found. And because we cut with EcoR1 and BAMH1, we have these sticky ends. And here is our plasmid, if it'll come up. Here is our plasmid. And it is cut with the same enzymes, EcoR1 here and BAMH1 here. And because of that cutting, we have complementary sticky ends. So we've cut the plasmid at its EcoR and BAMH1 site. Again, these sites are in the multiple cloning region. And the sticky ends are perfectly complementary to our gene of interest. They're perfectly mirrored our gene of interest. So the adenines and thymines are going to base pair on this side. The cytosine, thymine, adenine, and guanine are going to base pair on this side. And it's going to fit together perfectly only in this one orientation, the orientation that we want. It's impossible for this fragment to clone in the opposite orientation because the BAMH1 sticky end on this site doesn't match the EcoR1 on the plasmid, nor does this EcoR1 match the BAMH1. So our gene of interest fits perfectly into the multiple cloning site right here, and a little bit of ligase will seal the nick. And we can also reintroduce the idea of agarose gel electrophoresis here, because you can see that if we're cutting our gene of interest, we're also going to have these little scraps of DNA left behind that we're not interested in. And if we're going to cut away this middle region of our plasmid, we are going to have a little bit of a scrap region there. So we isolate the DNA that we wish to clone based on its size by running a gel, and then we physically extract the DNA out of the gel, isolate that DNA, and we can mix it together for a cloning reaction. So the two bands that we see here on this gel from the vector represent a very, very large band, a very, very long piece of DNA. That's the entirety of the plasmid, and a really small fragment as well. That small fragment is this little scrap piece from the initial plasmid that was liberated when we cut with echo and BAM. And when we run our gene of interest on a gel, we see a fairly sizable long DNA region of interest. That's our gene of interest. And these two little scrap pieces are the two side nubs that we've cut off as scraps from our gene of interest. So we would physically get a razor blade, we would run this gel, visualize it on a UV light box, and we would physically cut this band out of the gel with a razor blade. We would cut this band out of the gel with a razor blade. We would melt the gel down, liberate the DNA, capture the DNA based on its negative charge, and then once we isolate that DNA, we would mix it together in a single tube. Our gene of interest would anneal into the sticky ends that it is meant to in the plasmid, and we would add a little bit of DNA ligase to make those new phosphodiester bonds and create a whole entire plasmid, a recombinant plasmid. So just to zoom in a little bit on that multiple cloning site, that region of the plasmid that we clone this DNA into, this is what I mean by saying it's chock full of restriction enzyme sites, this all-purpose Swiss Army knife cloning region. It's just back-to-back restriction enzyme sites: ECHO, SAC, KPN, SMA, BAM, XBA, SAL1, PST1, SPH1, HINDI3, just one right after the other restriction enzyme sites, so that you can basically use whatever site is convenient for you. So in this case, we're just cutting with one enzyme, EchoR1, and we're going to liberate our genomic DNA using an EchoR1 digestion. We'll cut the plasmid at EchoR1. That's going to cut the EchoR1 site that's in the polylinker, and that allows us to clone one of these fragments into our plasmid. We'll seal it all up with DNA ligase, and we've got a recombinant plasmid. So that's how we make recombinant DNA. And what I wanted to end this lecture with was two examples of what we might do with recombinant DNA. And to be fair, only the first example actually requires recombinant DNA. The second example of what we'll talk about today is a kicked up version of monitoring gene expression, and it introduces a subtopic that I would like to explore not only today but also in our next session, called systems biology. But first, reporter genes. So reporter genes require the very types of techniques that we've been talking about in these first two lectures. It requires restriction enzymes, it requires cloning, it requires the creation or construction of recombinant DNA. And what reporter genes allow us to do is measure or determine when and sometimes where a specific gene is expressed. This is important for so many different reasons. In a multicellular organism, we might be interested in the spatial regulation of a gene. In other words, is it expressed at the extremities? Is it expressed in the nervous system? Is it expressed in specific tissue types? And the when is it expressed is also important temporally. So only in development, later on, etc. And in simpler organisms such as yeast, we can get a very good idea about what different environmental stimuli might cause these genes to turn on or turn off. So reporter genes have been around for a long, long time, and despite that, they remain just as relevant today as they were when they were first developed, because they answer some very, very important questions about when single specific genes are expressed. So oftentimes when you're trying to determine the role of a particular protein in the cell, the first question you ask is, well, I can learn about the role of this protein if I understand when and where the cell wants this protein. Another way to say that question is, when and where is the cell turning the gene for this protein on? That's a gene regulation question. To say it simply, when is this gene expressed and where is this gene expressed? So for example, you might think you have a gene that encodes a protein needed for proper neuron survival in the adult brain. You find, however, that it's expressed exclusively in the kidneys of a developing fetus. You've just proven your hypothesis incorrect. By determining where and when that gene was expressed, you have to reconsider your hypothesis. There's no way this protein is involved for proper neuron development in the adult brain if you see it only expressed in developing fetal kidneys. So we can answer some really simple but critical questions using reporter genes simply by determining the expression pattern or the regulation pattern of a gene. So, once again, a gene's expression pattern is almost always exclusively determined by its promoter, its regulatory sequences, and its transcription. We made that point in Unit 1. Almost all genes are regulated at the level of transcription initiation for energy efficiency. So if we can measure that regulation of transcription initiation using reporter genes, we can answer the question of when does the cell want this gene on. So in its simplest consideration, to make a reporter gene, we simply take the coding region of our gene of interest, we remove that, and we replace it with a coding region for a protein that's much easier to visualize, much easier to measure. That's essentially all a reporter gene is. So again, we have regulatory sequences in the promoter of our gene of interest. These are the sequences that control the expression of our gene of interest. That's what we're interested in measuring. When and where is our gene on? In other words, when and where are these promoter sequences active? And all of this is just the ATG to the stop codon. This is the instruction for building our protein of interest. We don't really need this to understand a regulation question. We can replace this with a reporter gene. We can replace this with the coding region for a gene that's much, much easier to detect and measure. And now our gene's promoter will control the expression of this visible protein. And when we see this protein expressed, we'll know that our promoter was on. And when we don't see our protein expressed, we'll know that our promoter is off. So we can use a little bit of an analogy here. Let's say that we have a thermostat in the wall, right? This represents the promoter. This is the control sequence. And we want to see, what is this thing controlling? We see it on the wall. It's got numbers on it. Sometimes these numbers change. Sometimes we hear a click from this thing, but we really don't know what's controlling this thing. We don't understand the environmental stimuli or the environmental conditions that cause this thing to change. And that's very much the kind of question that we ask in molecular biology. We know the promoter is there. We can see the promoter just like we can see this device on the wall. But we don't understand what promoters are responding to, what different conditions in the cell are turning the gene on or off. Now, we can track the wire that comes out of this device and it leads into the basement, and when we go into the basement, we see this thing. We don't even know what this thing is. This is just huge. I can't determine what this does. I can't understand what it is that I'm looking at. Just like the complexity of a gene that we find, right? We can track the wire between the promoter and the gene. The gene is the first ATG, or it starts at the first ATG right downstream of the promoter. So that's easy to find. But just by looking at the gene, we don't understand what it does. Even if we can decode that genetic sequence using the genetic code and determine the amino acid sequence of the protein, that still doesn't tell us what that protein does. It's largely a black box, a mystery. So we can get clever. We can say, okay, let's cut this wire so that now this thing, whatever it is, controls something else. And let's put at the end of that wire a light bulb, something that's very easy to detect, observe, and measure. And what we notice is that when the temperature in our room is very, very high, that light bulb stays off. When the temperature in our room gets quite cold, all of a sudden the light bulb turns on, and we say, now I understand. It's temperature that's controlling this switch. This device is responding to temperature. So I bet you that beast, that monster, that behemoth in the basement must have something to do with heating the room. Because when the room gets cold, this control device tells that machine in the basement to turn on, just like it told our light bulb to turn on. And when the temperature in the room is very, very high, this control device tells our machine in the basement to turn off, just like the light bulb turns off. So by rewiring this control mechanism to something we can easily detect and see, we can begin to deduce what controls our gene of interest. So again, just to tie the analogy into what we're viewing, the control region here, the promoter of the gene, is the thermostat. It's controlling our gene just like the thermostat controls the boiler in the basement, but we don't know what our gene does yet. We don't know what the boiler in the basement does yet, and we don't know what our control region is responding to, just like we didn't know what the thermostat was responding to. By taking the coding region and replacing it with something we can easily detect, like the light bulb in this analogy, now we can easily measure when the promoter is on and when the promoter is off, and we can begin to deduce what those signals mean for the cell and start to create informed hypotheses as to what the gene is actually doing. So we can say, aha, the gene is controlled by oxygen availability, and we can start to plan experiments around that hypothesis. So there are some common genes that are used as a reporter gene. Again, the reporter gene is the light bulb, the thing that we can easily see and detect. GFP is one of them. GFP stands for green fluorescent protein, and being a protein, it has a gene that encodes it. GFP, when expressed as a protein, fluoresces green under UV light. And what you're looking at here, this beautiful, beautiful image, is a C. elegans worm, a transparent microscopic worm, flatworm called C. elegans. And its neurons have been stained with GFP. That staining is through recombinant DNA technology. A neuron-specific protein has been linked to green fluorescent protein so that every single neuron is expressing this GFP, and that was done as a reporter gene. That was done by fusing the GFP gene to a promoter that is neuron-specific. So this protein, again, when expressed, glows green under UV light. Another reporter gene is the gene for the enzyme luciferase. We've talked about luciferase already. Luciferase plays a major role in second-gen sequencing. In the presence of ATP, luciferase will give off light, and this, again, is what fireflies express in their thorax, their abdomen, their lower region, to light up. So we see here flasks of luciferase in the presence of ATP giving off visible light. So here, if we see light is being made, that means the luciferase enzyme is around. If the luciferase enzyme is being expressed, that means the promoter is active, and we've just understood what's triggering our promoter. The promoter of our gene of interest is the thermostat in the wall, and the luciferase here is the light bulb we've rewired it to so that we can easily measure and detect when our promoter is active. So another way to determine the when and where of gene expression is a technique called in situ hybridization. But in situ hybridization goes a bit beyond the scope of this course, a little bit beyond reporter genes and what we'll talk about next, so we won't discuss it here. But I'm happy to go over in situ hybridization with you one-on-one if you have an interest or a curiosity there. All right, so let's move on to and close with microarrays. Microarrays, much like reporter genes, allow us to answer the question which genes are on or off. But here, this is a genome-wide gene expression experiment. Reporter genes allow us to do single gene studies. We've taken a single promoter from a single gene, we've fused it to a single reporter gene, and we're just asking questions about when is this one gene on and when is this one gene off. Microarrays allow us to address the question what all genes across the whole genome are on or off under different conditions. This idea, again, of systems biology, understanding the entire organism as a single living system, something we'll return to in the last lecture of this unit. So reporter genes and in situ hybridization are great, but they are for single gene studies. You, as the experimenter, have to manually create this recombinant DNA molecule that connects the promoter of your gene of interest to the reporter gene and interrogate the cell with that one artificially made reporter construct. It would be nice, indeed it would be very, very powerful, to get a snapshot of all the genes that are being expressed in a cell at any particular time and comparing that to all of the genes expressed by the same cell under different conditions. We can do this with a specific mRNA library called a cDNA library, but it's not really easy, for one, and it's certainly not fast. Microarrays allow us to answer this genome-wide expression pattern question very, very quickly and very effectively. It was invented in the late 1990s. Actually, this was invented while I was a grad student, so I saw this just emerge and explode onto the molecular genetics scene. DNA microarrays allow you to truly detect every single gene that is being transcribed by a single population of cells, period, across the whole genome. That means we're looking at and detecting the presence of every single messenger RNA made by that cell. And we get from this a very good idea of how much messenger RNAs were made in that time as well. So we get some kind of crude semi-quantitative idea of which genes are expressed at a high level, intermediate level, and low level. Most commonly, a DNA microarray is a glass slide that has been created by robotics. It's divided into a microscopic grid, so you can almost think of it like graph paper. And each square grid, each grid square of that molecular graph paper, of that etched glass microscope slide, has in it identical single-stranded DNA molecules that are adhered directly to the glass. Now, this is what's key. Even though each individual grid square has many, many identical DNA molecules in it, each grid square has a different set of molecules than any other. And essentially, what's happening here is that each grid square has DNA in it that represents the sequences of a specific gene. So what we're doing here is we're zooming in on, in this case, 6 particular grid squares. This grid square here contains many, 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 hundreds if not thousands of single-stranded DNA molecules, again adhered to the glass, and each one of those contains the sequence TGGGG. 2 grid squares over, we see another grid square, another region of this microscope slide, that contains thousands of single-stranded DNA molecules with the sequence ATCAG. Another grid square, GAGCT, another grid square, CAGTC. So each grid square contains identical DNA molecules within it, but different sequences than neighboring grid squares. And again, every single grid square represents the sequence of one specific gene from the organism that the microarray is created for. So in this way, one particular messenger RNA could bind to the single-stranded DNA found in the grid square that corresponds to the gene for that messenger RNA. And it would only bind to that particular grid square because that same sequence wouldn't be found anywhere else. So hundreds of thousands of different DNA sequences can be represented on a microarray, representing hundreds of thousands of different genes, and allowing the messenger RNAs to bind to them and detect their expression, detect that gene's transcription. What makes this powerful is that, of course, the grid square location of each particular sequence is known, so where every single gene from the genome is represented on the microarray is known as well. In other words, if you think of this as kind of a specific coordinates, almost like battleship, row C, column 18, is the ADH1 gene. We know that that gene's sequences are represented in that grid square. Z43 represents the AMB1 gene, and we know that that grid square represents the single-stranded DNA from AMB1. So, how does this all come together and work? We start by making cDNA. cDNA is complementary DNA, and what the cDNA is complementary to is messenger RNA. So mRNA is isolated from 2 different cell populations. Let's say, for the sake of example, cancer cells and healthy cells. So we might have healthy lung cells in one population and cancerous lung cells in another. We lyse those cells open and we extract and isolate the messenger RNA from all of those cells. And then we reverse transcribe that messenger RNA to cDNA. By reverse transcription, we mean going against the flow of transcription. Transcription is copying DNA to RNA. Reverse transcription is copying RNA to DNA. We can do this using specific viral enzymes called reverse transcriptases. It doesn't naturally happen in most of the natural world, but a specific form of virus does do this, and we can leverage its enzymes and use these reverse transcriptases to do this. So we've isolated mRNA from healthy cells, and we've reverse transcribed it into DNA. We've isolated messenger RNAs from cancerous cells, and we've reverse transcribed it to DNA as well. When we do this reverse transcription, we use specific fluorescently labeled bases for each population. For example, the cDNA from the healthy cells might be done, that reverse transcription might be done in the presence of green fluorophores. So now all of those cDNAs fluoresce green, while the cDNA from the cancer cells could have been reverse transcribed in the presence of red fluorophores, so all of those cDNAs will fluoresce red. So what we're left with after this process is two collections or populations of cDNAs. The cDNAs that fluoresce green are DNA copies of all the messenger RNAs from all the genes that were active in the healthy cells. And the cDNAs that fluoresce red represent DNA copies of all the messenger RNAs of all the genes that were active in the cancerous cells. Now we heat that DNA up to make it single-stranded, and we add that single-stranded cDNA to the microarray, all of it, the reds and the greens. The cDNA, because it's complementary to the messenger RNA, is also complementary to the genes represented on the microarray, and the cDNA will bind to any of that single-stranded DNA found on any microarray grid square that it's complementary to. Remember, each grid square represents one gene. So any gene that was on in the cell's population had messenger RNA for that gene transcribed. We've made cDNA to that gene that fluoresces, and now that cDNA can anneal to and bind to that grid square. This is called hybridization. If a particular grid square is bound by cDNA from healthy cells, that grid square is going to fluoresce green because that's what that cDNA is doing. And of course, cDNA from cancerous cells will fluoresce red. And that's going to show us that that gene was present in cancerous cells. If a particular grid square is bound by cDNA from both healthy and cancerous cells, it fluoresces yellow. In fluorescence, when red and green overlap, we see that as yellow in fluorescence. And not surprisingly, if there is no cDNA present for any particular gene, then that grid square will not bind any cDNA and it will remain black. So yellow grid squares on a microarray represent the presence of messenger RNAs that both cells were making. Cancerous cells were making this messenger RNA. In other words, those genes were on in cancerous cells, and those genes were also on in the healthy cells. Red glowing grid squares represent genes that were expressed only in cancerous cells. This is a characteristic of genes called oncogenes, genes that tend to encode proteins that promote cancer growth. And then grid squares that glow green represent genes that were only being transcribed in the healthy cells, not in the cancerous cells. And that represents tumor suppressor genes, genes that encode proteins that keep cancer at bay. And again, black squares represent genes that weren't expressed in either population. So, by looking at the expression profiles that we see, and looking at what genes are on and off in healthy and cancerous cells respectively, we can begin to understand what genes are doing and the proteins they encode, what roles they're playing in tumor progression. We're looking at the entire genome in one snapshot and getting a wealth of information about the genes that are on and off. And remember, I mentioned it earlier, but the intensity of the color is also meaningful. So this is a gene that is expressed in healthy cells and expressed at a very high level. This is a gene that's expressed in cancerous cells only and expressed at a very high level in cancerous cells. This is a gene that's expressed in both cells at a high level. This is a gene that's expressed at low levels in healthy cells. This is a gene that's expressed in low levels in cancerous cells, etc. So we're looking, I can't say it enough, we're looking at every single gene that's on and off in two populations of cells across the entire genome at any one time. Pretty incredible. Again, because you know which genes are represented by which grid square, you instantly know the identity of these different genes, and you can begin to ask true scientific questions about why. Why are these genes being differentially expressed? What is their role in cancer protection or cancer progression? Truly amazing. Microarrays have helped us identify truly tons of genes important for so many different things. For example, genes that are needed for the ripening of fruit were identified through microarrays. Not surprisingly, genes that are needed for cancer protection or progression, same thing, using the same kind of experimental approach that we just described. In fact, cancerous tumors are now profiled individually for their unique gene expression signatures. This is called personalized medicine, and this helps doctors determine how aggressive the tumor might be, predict the effects of different therapies, and tailor a treatment to a specific tumor type or class, and give patients the best prognosis for recovery because these individual tumors are being treated as individual diseases rather than a one-size-fits-all breast cancer or prostate cancer. So just as an example, we biopsy the tumor, we do microarray analysis on the tumor, we look to see all of the genes that the tumor is and is not expressing, and based on that genomic surveying of gene expression in that tumor, we know that we've got either a non-responding tumor to particular treatments, we won't even go down the road of that treatment, or we have a responder tumor to that treatment. We know that that treatment is going to have a high degree of efficacy against that particular tumor. So we're truly treating the disease the patient has rather than the disease we think it falls into. Amazing stuff. So hopefully I've passed on just some introduction to this. We're going to come back to systems biology in the next lecture next week. But to summarize what we talked about here, recombinant DNA molecules, again, DNA that is not naturally occurring and is artificially made in a lab, can be created by taking two different pieces of DNA and ligating them together, usually in the context of a plasmid. This has ideas of cloning, directional cloning, when we use two different restriction enzymes to ensure that our insert is going on in the proper orientation or direction. And we touched on restriction enzymes and plasmids as well. For reporter genes, we take the coding region of the gene of interest, get rid of that, and replace it with the coding region of a detector or reporter gene that's much easier to see. And this allows us to measure the expression of that one gene in any context or condition that we wish. That allows us to study the expression of one gene at one time. We also have DNA microarrays that take that idea but expand it and allow us to see the expression of every single gene in a cellular population at one time. We do this by isolating messenger RNA, reverse transcribing it into fluorescent cDNA, denaturing or melting that cDNA into single strands, and then adding that to the microarray. The microarray itself is a grid system with single-stranded DNA probes representing every single gene. Each grid square gets its own gene, and where these fluorescent cDNAs bind and anneal due to their complementarity causes that grid square to fluoresce, and we can detect the presence of that cDNA for that gene, which correlates to the messenger RNA for that gene, and we know that gene was being transcribed. So awesome stuff. I never get sick of it. Hopefully you have some appreciation for its awesomeness as well. You know what to do when the dialog box pops up. Let me know what is still confusing, and we will review those concepts. We'll explore some deeper critical thinking challenge questions, and we'll talk about some papers in this arena when we meet in person on Friday. Until then, thanks for watching, and I'll see you all soon.